Alright students, so after you all left, I realized that the recording cut out right after I started talking about cyclin, and so I want to make sure to record that discussion. Just follow along to refresh your memory. So cyclin, in response to DNA replication and also that growth signal, starts being produced in the middle of the S phase. By the time they get to G2, cyclin has accumulated and starts to associate with something called CDKs, or cyclin-dependent kinases. And when these cyclin-dependent kinases attach to the cyclins, that is referred to as an MPF, or M-phase maturation promoting factor. And so as you can see right here, there is cyclin attached to CDK in this complex. And as that happens, CDKs are always present in your cells, and so the concentration of MPFs starts to increase as you get closer to the G2 checkpoint. And that's essentially what makes the cell go from G2 into M phase, is the concentration of MPF. Now, what I want to talk about is also what I mentioned with the 4 phases of mitosis. Right? So after passing that G2 checkpoint, the cell will enter our prophase phase of mitosis. And since we've already gone through the S phase by this point, I mentioned that our chromosomes will already have been replicated. And so what we see here, this is chromosome 1, maternal and paternal copy, is that we would expect to see sister chromatids for each copy, right, thereby forming a tetrad. And so in prophase, we start to see these chromosomes that have been duplicated become very, very clear inside the nucleus. And in addition, I mentioned that this organelle called centrioles, they start to become more prominently seen, and they actually replicate, so we have 2 of them. Now these centrioles are going to be really important because they produce something called microtubules, which are going to be used to attach to each of these chromosomes. And so after we go through prophase, we enter metaphase. And in metaphase, the characteristic of metaphase is that the nucleus, you can see, has dissolved, and the chromosomes, the homologous chromosomes, they line up along the metaphasal plate in preparation for division. The centrioles, if you've noticed, they've migrated to each pole of the cell, and these dotted lines were meant to represent microtubules being synthesized by the centrioles. Microtubules, again, are an element of the cytoskeleton. There's 3 types of molecules that are synthesized in the cytoskeleton. Microtubules are one of them. And as these microtubules extend, they actually attach to the centromere of these replicated chromosomes. And when that happens, we call that a kinetochore. That's the microtubule attached to the centromere. And if you were actually to zoom in even more, there are these little proteins that are going to literally pull the chromosome to either side of the cell. And as they do that, they actually walk along the microtubule. It's pretty cool. So we have metaphase. Now something that I mentioned in class that I want to clarify here is that actually a checkpoint within the mitotic phase, it happens here. I incorrectly stated that the checkpoint was this double checking for a clean division. This actually happens right about here. During metaphase, what's being determined is if all the chromosomes have attached to a microtubule, right? Because it cannot proceed to anaphase unless those kinetochores are all ready to go, right? So the big M checkpoint is actually during metaphase, okay? All right, so now we have our kinetochores formed. What's going to end up happening is that, like I mentioned, at the kinetochores, you have these little motor proteins that will literally pull the sister chromatids apart and walk the chromosomes towards the centrioles. And as that happens, you actually see the microtubules disintegrate after the motor protein has passed it by. And so this pulls apart the two sister chromatids until they are separated into the two poles of the cell. Now, at this point, what we begin to see is that the chromosomes are now within the two sides of the cell, and we see nuclei start to reform. And inside, we have our chromosomes. All right, so we have our two chromosomes, and what is able to happen here is that we start seeing, as these nuclei form, that the cell membrane begins to pinch inwards, creating something called a cleavage furrow. Right, and what comes to completion is in cytokinesis, right? So we have our nuclei formed, we have the beginnings of the cleavage furrow. Cytokinesis is simply the complete division of the cytoplasm after telophase. And so you have two distinct cells, right? And you can kind of imagine what those would look like. I'll draw a small version of each of those. Is you'll have two what are known as daughter cells. Each with their copy of the chromosome. Now, these daughter cells are going to be genetically identical to the cell that preceded prophase, right? Because these chromosomes entering prophase have already been duplicated. The cell that was there in the G1 phase would have only had one maternal and one paternal chromosome 1. So we get identical daughter cells out of mitosis. Okay. So going back to our objectives for this, we wanted to talk about chromosomes, talk about the role of mitosis, talk about the cell cycle and the stages of mitosis. I hope that was helpful, and please let me know if you have any questions, and I'd be happy to go over more with you in class. Thank you.